So for the last section today, we got done talking about immunoglobulin gene rearrangement last time, and we saw how much variability can be made in the different antibody responses to different antigens that will be presented, or different foreign objects that will be presented to the host system. And so this time we want to take a look and see just how that antigen is actually presented to the B and T cells so that you can start generating an antibody response. And so this is going to involve the major histocompatibility complexes, and there's two main ones. There's MHC1 and MHC2 pathways. So if we take a look at our cell, these are going to be the antigen presentation pathways. So if a bacteria comes into the cell, the bacteria will get engulfed typically, or at least some of the bacteria get engulfed by our innate immune response. So either dendritic cells or natural killer cells or macrophage are going to recognize the bacteria as being a foreign substance coming into the body, and they're going to engulf or phagocytize the bacterium, and they start to break it down. And so once they start breaking down the bacteria, some of the proteins that are on the surface of the bacteria make up the coat protein actually will get chewed up, and they get recognized by our major histocompatibility complex pathways. So there's two major ones. There's MHC1 and MHC2 that we're going to talk about. So MHC1 occurs when you have endogenous antigen that is degraded by the proteasome. So this cell has phagocytized a bacteria or other foreign invader, and now that it's entered into the cell, it's starting to break down these components in the proteasome. And what happens then is that these degradation products are going to come and they're going to enter into the endoplasmic reticulum, which is that lipid bilayer area that's connected onto the nucleus of the cell. So you can see here's the nucleus of the cell, and so we're inside here, and we're going into the endoplasmic reticulum now. And so the endoplasmic reticulum is where most of the P450 enzymes actually reside within cells to mediate the detoxification reactions of compounds that are going to come into the cell. It's also a place where the lipids are going to be synthesized, and where molecules that need to go out of the cell or into the cell membrane are going to be processed. So the endoplasmic reticulum actually has many multiple functions within the cell. In immune cells, like the natural killer cells or the macrophage or the dendrite cells from the innate immune response, they have the major histocompatibility complex proteins that are embedded inside the membrane of the endoplasmic reticulum. So the peptide antigens that are coming from the surface proteins of the bacteria that got engulfed and got eaten by this cell, pieces of these peptides are going to get transported inside the endoplasmic reticulum. So it's going to be transported through this TAP protein, which is a transporter associated with the antigen protein. So it's kind of a wordy nomenclature for this protein, but you can just call it the TAP protein. And just remember that its purpose is to start transporting these small peptides that get chewed up from the proteasome into the endoplasmic reticulum, where they can be activated to start interacting with our major histocompatibility complex in pathway 1. So they'll enter into the endoplasmic reticulum, and the MHC then, the alpha chain, is going to bind to some initiator proteins. So there's other proteins inside the endoplasmic reticulum that need to kind of activate the major histocompatibility complex so that it can interact with this antigen that's coming in. So you don't need to know the names of these different proteins, but one is called calnexin, and then there's a beta-2 microglobulin that also will bind to and associate with this complex. The calnexin will then dissociate once these two guys have bound, and then the calreticulin and the tipazin actually will bind as well. So there's two more proteins that will come in and they'll bind, and these guys are actually just acting as cofactors. They help this little small peptide that comes from the bacteria or other foreign invader they're going to help it be recognized by this protein so that it can bind to it. These guys are then going to fall off, and the major histocompatibility complex then is going to be moved then to the cell membrane where it can be exposed on the surface of the cell. So this is going to be the antigen presenting process so that these cells can actually interact now with B and T cells so that you start getting the antibody rearrangement process going on in the B and T cells, and you get the adaptive immune response started to the actual invader that has come into the cell. So the B and T cells would then start to make antibodies that would be very specific for the invading organism. So this is just showing those auxiliary proteins coming, binding to the major histocompatibility complex, and they cause the association of this peptide. It will then form in a secretory vesicle, get transported to the Golgi apparatus, and the Golgi complex, it'll fuse with the Golgi complex, and further processing will take place that's required to actually transport it to the plasma membrane. So it goes through here, gets further processed so that it gets docked right into that membrane, and then it fuses with the plasma membrane, and now the antigen is being presented in association with the major histocompatibility complex 1 on the surface of the cell. If we look at the process for the major histocompatibility complex for number 2, we'll see it's just a little bit different. So our major histocompatibility complex 2 is actually bound to an invariant chain, so it's got another peptide linkage in here that actually blocks the binding of endogenous antigen. So the antigen that's come in and it got digested by the proteasome is not going to be able to bind directly to the major histocompatibility complex 2. You have to have further processing of this protein to allow it to bind to actually exogenous antigen. And so if we follow the processing of 2, it will come out and it will form a secretory vesicle, and it's going to get routed through the Golgi apparatus just like our other major histocompatibility complex did, and it's going to get clipped at this point, and this leaves a little tiny clip fragment in the binding site where the antigen will need to bind to. So at this point, exogenous antigen from the outside is taken up, and it will be degraded and routed to the endocytic pathway compartments. And so it will fuse to this secretory vesicle, and it will allow the replacement of that little clip fragment into the antigen binding site. And so this antigen will then bind to the major histocompatibility complex, and this will get transported out to the plasma membrane as well. And so this guy is also presenting antigen at the surface of the cell. And so by having both of these classes of compounds and having a little bit different approach for isolating and gathering the antigenic information from the invading bacteria, one coming from the phagocytized bacterium, the other coming from an exogenous source that's outside the cell and not being completely digested, is going to give different fragments the opportunity to become antigens. And so you'll have a better immune response if you have multiple antigen fragments that get presented onto the surface of these cells. So you can come back and go through here and you can do the comparison pathway as well. So if you just come to the Stanford website and take a look through here, you'll see how these helper cells, or the innate immune response, is going to be responsible for presenting antigen to our antibody producing cells within the immune system, the T and the B cells. Alright, so now that we've talked about the major histocompatibility complex in a little bit more detail, we're going to talk about the mechanism of antibody formation in our primary immune response. Okay, so this is going to be the formation of very specific antibodies. So this is our adaptive immune system that's going to start to build antibody production against an invading pathogenic microorganism. So this is called humoral immunity. So antibody formation to foreign compounds or foreign biological components, so about 90% of these, which are the antigens then, adhere to this mechanism of formation or antibody response. So these antigens are called T cell dependent because the mechanism for antibody formation actually requires T cell help. So the participating immune cells that are required for this antibody response are going to be our antigen presenting cell. So again, this is what we were talking about in the last little section, talking about our dendritic cells or our natural killer cells or the neutrophils or the macrophage in our innate immune response that basically are going to eat up the foreign invader, the bacteria, and start presenting antigen from that organism on the surface of their cell in conjunction with the major histocompatibility complex. So the other cells that we haven't talked a lot about yet are the T and the B cells. So just from the hematopoietic standpoint, we talked about their production, but we didn't talk about exactly how they get activated within the immune system. So we'll take a look at how this process occurs and how these cells start to dedicate their antibody production through genetic rearrangement, so rearranging their chromosomes to produce very specific antibodies. So this is going to require a T helper cell, or a TH, the B cell, and then the B cell as it transforms and commits to becoming a plasma cell. So if we have a microorganism, like in a bacterium, this can immunologically be considered as an assortment of different antigens. So all these different proteins that are on the surface of the bacteria can actually cause an immune response. So you might have some epitopes that could be found by either T cells or B cells in all of these different fragments. And that's part of the miracle of our T and B cells. So each of the different antibody cells that are floating around in our bloodstream, they can recognize just a little bit different antigens that get presented from our foreign surfaces. And so they kind of have recognition motifs. Like it would be like having a bunch of different cookie cutter shapes on the edge of the antibodies that are going to be detected by the T and the B cell precursors that are floating around in the bloodstream and haven't fully differentiated at that point. So they're looking for foreign invaders, and once they find an antigen presenting cell that's going to take and degrade these types of proteins from the bacterium and then combine it with the major histocompatibility complex proteins and then display it on the surface of the cell, it can actually interact then with the antibodies and it can start generating an immune response. So we'll take a look at this as far as our dendritic cell is going to be the cell that comes and it's going to engulf the bacteria. So it undergoes phagocytosis and it encloses this now in a vesicle. This is called a phagosome. And so our bacteria is inside. It's going to fuse with the lysosome, and all of these lytic enzymes are going to come and they're going to start degrading the bacteria. So there's proteases in here, there's lipases in here, there's enzymes that will break down carbohydrates, and they basically break down the bacteria into its small components or pieces. So a few of these peptides that would be present in here are going to be T cell epitopes. And so they're the ones that are indicated in red on this slide for our demonstration. So here's our MHC class 2 protein. So this would be an exogenous example. It's going to release the clip fragment and now it's bound to one of these epitopes. So this is going to get moved to the surface of our dendritic cell for our major histocompatibility complex 2. So now it's got antigen presenting, MHC associated antigen presenting capabilities all over the surface of the cell. So it does this multiple times for the bacteria that it is engulfed. So then when a T helper cell comes along, it's got a recognition protein that's going to actually recognize the major histocompatibility complex. So here's our T cell receptor. There's a CD4 protein that's going to help recognize the major histocompatibility complex. And the docking station over here is going to help line up and recognize this antigen. So this specific T helper cell is very specific for that antigen that comes from that foreign bacteria. So at this point, if it binds to an antigen on a dendritic cell or an antigen presenting cell, it's going to start releasing cytokines like interleukin 2, and it also has receptors to the interleukins that can start and activate pathways within this T helper cell. So the binding of interleukin 2 to the interleukin 2 receptor is going to initiate cell division. And so you've got clonal expansion now that starts to occur with your T helper cells. And this is good because this is the specific T helper cell that recognizes the antigen that is present on the bacteria that are now infecting this host. So it's starting to wage warfare, essentially, by mass producing the T helper cells of our immune system that are going to recognize that foreign invader. So a particular B cell is also going to have specificity for the B cell epitope, and it's going to bind to the antigen through the B cell receptor. So B cells have antibodies that are actually exposed and being produced on the surface of the cell, and they basically antibody B cell that can recognize every different epitope that is out there. And so the B cell is going to recognize foreign proteins just like this, and it's going to bind to these, it's going to engulf them, and it's going to digest them, just like we saw with the dendritic cell, except it does this for a single protein. It can also then transfer portions of the antigens to the major histocompatibility complex 2 proteins and remove that little clip fragment, and then it's going to associate the MHC2 class onto the surface of the cell. So now you've got this B cell. It has antibodies that are going to recognize that specific epitope from the bacterial invader, and now it's also already digested some of that protein, and it's presenting the antigen in association with the major histocompatibility complex 2 on the surface of its cell as well. So our T helper cell now that recognizes that antigen is going to bind to this cell, and then this one, so you get this same kind of interaction. You've got a bidirectional activation now that's caused by the T cell receptor to the T cell epitope of the MHC class 2 interaction. So when these cells bind together, when the T helper cell docks down here, this causes a response in the T helper cell so that it's going to express and secrete interleukin 4 and interleukin 5. So the T helper cell is going to make more cytokines, and it's going to release them right in this area. The binding of the T cell receptor to the B cell is going to cause the B cell to actually express and place on its surface receptors for interleukin 4 and interleukin 5. So it's going to express the receptors. The T helper cell is going to express the interleukins that are going to bind to those receptors, and you'll get an even further stimulation and activation of this B cell. So you'll see this interaction of the 2 proteins. You start seeing the accumulation now of the interleukin 4 and interleukin 5 receptors on the surface of the B cell. The cytokines, IL-4 and IL-5, are going to be secreted from the T helper cell. They're going to bind to these receptors now, and they're going to activate the B cell. So the binding of the IL-4 and IL-5 to the B cell causes the B cell to start to replicate itself and divide. So you get clonal expansion of this particular B cell that is producing antibodies that recognize the foreign invader. So you start building up your platoon of B cells that are going to be able to fight off the infection for the specific bacteria that is infecting the cell. So some of these B cells are going to differentiate into plasma cells, and these guys produce large amounts of soluble antibody that's going to be released. So the plasma cells that come from these B cells are going to express antibodies of the IgM class. And so if you actually look at a molecule of the IgM class, let's see if we can get it to secrete one again. Well, pause, stop. Stop. It won't pause while it's floating by. Okay, I'll have to like grab the IgM class really quick so that you guys can see this antibody class in a little more detail. So the type of antibodies that we looked at last time looked a lot like this. So our antibody shape showing the heavy chain connected with the light chain. The antigen binding region would be out here on the class of enzymes. So this would be typical of both the IgE and IgG antibody subtypes, that they have 2 heavy chains and 2 light chains per antibody. The IgM class, however, is a little bit different. And let me see if I can find a picture of the IgM class. This is a nice one. Okay, so if we click on this one, so this is just showing some of the different classes. Our IgE, IgG, and IgD come in the monomer form where they have the 2 light chains and the 2 heavy chains coming together. If you form a dimer between 2 of these monomers, you will get the IgA class of antibodies. And then if you form a pentamer, so if you've got this core structure down here that actually will dock 5 of these monoclonal antibodies onto a specific docking station, you get antibodies that produce 5 different separate antibodies, and each of these guys can recognize 2 antigens. So the binding capacity of a single IgM antibody is going to be able to bind 10 epitopes or 10 antigens that are surrounding there. And so what the immune system does when it secretes these antibodies into the bloodstream is anytime it finds bacteria, it's going to start sticking them together in big clumps so that you can actually recognize and get rid of them from the body. It makes it much less effective to get clumped together like that. So if we go back to our diagram, so our plasma cell is predominantly expressing this IgM isotype of antibody. Some of the B cells that get produced will also go on to produce memory cells. So once you have been infected with a specific bacteria, your immune system wants to remember that you've been infected to them. And so they actually will stockpile these memory cells, and they'll produce quite a few of them that will end up circulating or floating around within your system. And this is why we can develop immunity to specific bacteria. So the next time that you're infected by the same bacterium, it usually will not get a foothold because there's a number of these memory cells that are around. So instead of having maybe like 1 in a billion cells that actually recognizes that epitope, you now have 1 in 10,000 cells of your B cells actually recognize that epitope. So the next time that foreign invader comes in, it's got a much more highly likelihood that that memory cell is going to find and recognize that antigen very quickly and start mass producing the antibodies again so that you can fight the infection very quickly the second time around. Okay, so this is just a summary of the different components that you'll see involved in the immune response. Okay, so this last section that we'll talk about the immune system is, and unfortunately this is in another language, and I don't even know what language this is, but that doesn't matter. It's partially in English, partially in, it looks almost Latin to me, but anyway, okay, so we will skip doing the reading on this section, but I want to introduce you to the complement system. And the complement system is actually an important component that helps tie together the innate immune response with the adaptive immune response. So we've already seen that they talk to each other to get the adaptive immune response started. You need the innate immune cells to actually accumulate the antigen from the incoming infection. So whatever the foreign invader is into the cell, the innate immune system is going to collect that antigen through these dendritic cells or the neutrophils or the macrophage They kind of swallow it up and they will present it on the surface of their cells with the major histocompatibility complexes. So we've seen that going on. And this starts the adaptive response where the antibodies that are going to be very specific for those compounds get activated. So the complement system is a way that the adaptive immune response can now help recruit some of the innate immune response systems to make the innate immune system more effective at gathering up the foreign entities and phagocytizing them and then disposing of them in a very quick manner. And so there's the classical pathway for the complement system, the lectin pathway, and the alternative pathways that are active. But they all kind of work in the same general sense, and so we'll get an overview of the complement system. And you don't need to know the specific proteins that are actually involved, but basically I want you to know kind of what they're actually doing to help reduce or get rid of a foreign invader like a bacteria that might be in the host and causing an infection. So the complement system has 4 major functions in the way that it helps the adaptive immune response kind of recruit the innate immune response. And so the first one of these is opsonization. And so opsonization is listed down here on this slide. And this is actually the enhancement of phagocytosis by the innate immune cell responders. The production of the antibodies is actually going to help the dendritic cells, the neutrophil cells, the natural killer cells, and the macrophage to be able to recognize and bind to the foreign invaders and engulf them. And so so this property of opsonization is basically like optimization. You can kind of think of them as one and the same. It's going to optimize the ability of these innate immune cells to be able to engulf and destroy the infection from the body. There also is this process of chemotaxis. And so chemotaxis, we saw actually a very good example of this on YouTube with our neutrophil actually chasing around the bacteria and trying to swallow it up. So this process of chemotaxis is actually sending out little sensory molecules that can recognize the bacteria that will help attract macrophage and neutrophils to the bacteria. And so the neutrophils and the macrophage actually appear to chase the bacteria. And it's due to this chemotaxis property of these small molecules that get activated through the complement system that end up binding to the bacteria that are basically these little beacons or sensors that will attract chemically. So it's like they almost smell or have an odor to them that they send out, and they will attract the neutrophil and the macrophage to them. They will also increase the lysis or the rupturing of the membranes on foreign cells. So they will help break open the bacteria, make them more porous and susceptible to rupturing. And they also will help with clumping of the antigen-bearing agents. So what this means is that when you have antibodies that are actually sticking onto the bacteria, they will start to clump together. Like the antibodies have multiple, especially the M-class antibodies, have at least 10 sites where the antigens can actually bind. And so the antibodies that are associated with the IgM class can then bind to multiple organisms. So they can, bind up to 10 different bacteria. Typically, they probably won't bind that many, but you could see if you have, more than one antibody around that's been solubilized, you can get big clumps of the bacteria that get all tied up together because the antibodies are sticking to them and causing them to stick together. And so this clumping, it will decrease their efficiency in being able to attack and infect the host cells. And it also is going to increase the efficiency of the macrophage, the neutrophils, and the dendrocyte and the dendritic cells, because they're going to find a big clump of bacteria now to engulf and phagocytize and chew up and destroy, rather than having to search them out one by one. So the complement system is going to help this process along. So the complement system is largely composed of these proteins that are called proteases. Many proteins within the body get activated when they get cleaved. And so if you break them in half, they're like they're like stored in this inactive state, in a very elongated state, so they have more protein associated with them than they need to be active. And so they're held in an inactive state into the cell until they're actually required or needed, and then they'll get cleaved or cut apart, and this is when they become active. So the whole blood coagulation cascade pathway involves this series of zymogens that would be cleaved, and then following the activation, you would actually have a blood clotting event. Apoptosis within the cell, or cell death, programmed cell death, usually is mediated in this fashion too. You have many zymogens. Once they get activated, they start cleaving each other, become active, and then the proteases start destroying the proteins around the area and cause the cell death in that fashion. So in a similar way, the complement system is being run using these zymogen type of proteins that need to be cleaved or clipped off before they can become active. And once they become active, they become better at being able to aid the adaptive immune response and connect it with the innate immune system to help the dendrocytes, the macrophage, and the other innate immune cells to attack the foreign invader. If you've got bacterial infection in the bloodstream, you can see this is our IgM class of antibodies. We also maybe had some IgG class antibodies. And these proteins that are flying in here are the complement proteins that actually need to be cleaved then before they can become active. So you can see the cleavage of these guys starts to align them along the bacterial cell. So this is starting to bind and form clumpy regions on the bacteria that are going to be able to start sticking to each other. So they're forming sticky parts. And then this complement that also will start binding to the bacteria will start actually forming a pore structure that will start damaging the membrane. So you can see you've got recruitment, activation, you start cleaving more of the proteins. These guys become active when they're cleaved. This recruits some of the other complement proteins. So these are all small proteins that start coming together. So you can see they're given really interesting names like C3 and C5. They're all for complement. Complement 5, 6, 7, 8. Complement 9 comes in, and you start forming this huge pore. So it starts to break apart the membrane, and this is going to actually cause the bacteria to explode because you're breaking apart the plasma membrane of the bacteria. All that's going to be left is this little goo. And when the goo is left, the other cells can come and chew it up. The complement system also is going to have additional markers that our major innate immune responders, like the dendritic cell, are going to recognize. And so it can find the bacteria more easily and engulf it by phagocytosis, start to digest it with its digestive enzymes, and get rid of the bacteria. So complement will actually increase the effectiveness of our innate immune responders for actually catching and targeting this infection. So this is another example of the complement system working just a little bit differently. This one is the mannose-lectin pathway. And so you can see some of the IgG antibodies are recruited to this one. But you still are pulling in a lot of the complement proteins that need to be cleaved to become active. And so you're still recruiting all this complement to the surface of the bacteria in this case. And this is again going to help actually recruit either the dendritic cells or the macrophage or neutrophils. These also act as chemical signal molecules that help attract those cells so that they can actually chase the bacteria around and then bind to them and be able to engulf them. And so that process again is called chemotaxis. And so again, you don't need to remember all of the different complement components. Most of them are kind of given boring names like C3, C5, C8, C9. But you do need to remember that these guys are called zymogens, and so that's spelled Zymogens. And so these zymogens are proteins that have to be cleaved to become active. And basically, they're going to help The innate immune cells actually respond to an event, and this is going to be aided by the antibodies that actually can bind to these epitopes. They help the bacteria become more sticky so that they stick together in big clumps. That way our innate immune cells that are going to eat these bacteria can find them easier, and they can eat more of them at one time. So that's opsonization. And the opsonization is basically enabling the dendritic cells, the macrophages, the neutrophils, all the innate responders to become better at phagocytizing and eating the foreign invader. Chemotaxis is going to help attract those cells to the bacteria, which will also aid in the opsonization process. So all of these things really are enabling the innate immune responders to find the bacteria easier and to eat them up. So that is a basic introduction to our immune system. And next week, we'll talk more about hypersensitivity, and we'll talk about some of the defects in the immune system that can lead to disease states. So we'll also talk next week about inflammatory response as well, because that's all related to the immune system. And we'll start talking about our anti-inflammatory drugs and how we treat hypersensitivity or allergic reactions and some of the other disease states that can be caused by immune system defects.